And we're live. We were having a technical problem with Restream, and so since Dustin was trying to come over here and talk, um, we're trying StreamYard instead. So welcome. So we can talk Origins, go back to Origins questions and see if Dustin can get back in. So go ahead, speak your mind, Just Jackson.
About anything or about Origins?
Origins better.
Okay, yeah.
Because we
Derail
can.
here.
There are plenty of people to talk about politics, but
Yeah.
We can talk about Origins here.
Yeah.
By the way, I have to say, some of the people on John Maddox's channel I think are really rude, and I can't. And I felt kind of bad in some of the streams where it was like you're being interrogated, and
a little bit.
I thought it was way too confrontational, and these weren't even really good arguments either. So I did, my feeling is the way you get treated is I wouldn't want to be treated that way.
Yeah, well, thank you. I don't mind it though. I have thick skin. I enjoy the dumpster fires over there.
Some people enjoy them. I don't.
Yeah.
I really don't. And especially coming from the creationists who are on my side, I'm just like, you guys just don't represent. I don't think you're very good representatives of creationism. So let me at least say hi to some of the people here. So if you have questions, so nice to see you, Redeemed SWCM. Nice to see you. So thanks for joining. And I don't know why poor Dustin's not able to get on.
He's on his phone. But
he may have given up.
Yeah, I guess. But yeah, Dustin's not a fan of me. Maybe he's over that now. I don't know.
So, you had some questions. You were asking about genetic entropy one time.
Yeah, we were. Yeah, I got my notes over here somewhere. But yeah, I've got a few arguments I think against genetic entropy.
Okay.
Those equations I was doing. I've told you about that, right? Trying to equate a viral genome size and mutation rate with the human genome size and mutation rate, and kind of equating to see how long it's existed in equivalent to human. I said equivalent a lot in human terms. Is that making sense?
Yes.
Okay.
I could see what you're. Yeah, go on. I said it was a good attempt.
Yeah.
I don't weigh into that because there are people that actually did experimental research on the human genome and the mutation rate. So it's rather moot, even if your calculation is right. It's like superseded by our experimental observations.
Like how many mutations per generation?
Yeah.
Oh, I know. I took that into account, is what I'm saying. The 100 number.
Yeah, which is a very nice number because it makes all the calculations for us easier.
Yeah. Well, let me just read out the equation and see if it makes more sense. For smallpox, it's been around at least 3,500 years. It's 186,000 base pairs. Its replication time is like 7 days, I think I've said. I said per replication cycle it has 2 mutations. So 3 billion base pairs divided by 186,000, 16,129. Multiply that by 2 mutations, that comes up to 32,000 something. Divide that by 100 mutations, comes up to 322. Take 3,500 for 3,500 years, multiply that by 365, and then divide that by 7, the amount of times it takes to replicate. It comes out to 182,500. So 182,500 times 322 mutations is 58.7 million, or 58.7 million years. equivalent to in human terms. You follow what I'm talking about? I had to explain this to my dad. I was explaining it to him. I had to tell him like 15 times.
So it has, are you saying it replicates, you have more cell cycles and more mutations?
Let me see. Yeah, that's what I'm saying. Kind of, I think.
And my, what I've tried to point out is viruses are a terrible model organism to try to infer what's going to happen at the human level.
Yeah.
Because they don't have their own replication machinery, for one, and the other thing is humans are very sensitive to perturbation, and there are more things that can break because one, humans are multicellular, but there are all sorts of things. So I kept trying to tell people, I keep trying to tell people, don't equate the two. You can't even use bacteria as a good model organism because bacteria can lose 80% of their genome and it'll survive. You can't apply that to humans. I don't think any scientist would think we could lose 80% of our genome and still live.
Yeah.
In terms of the genes. Dustin, are you on? Are you connected? Is your microphone working? You can hear us, but I can't hear you. So, I think he's trying to get in.
Yeah.
So, am I getting your point, first of all?
I think so.
Okay. So my point is that I think you can show that viruses don't degrade like humans.
Yeah. But I know that John Sanford wrote a paper on H1N1 talking about
He
genetic.
did.
Yeah.
He did. And I had reservations about going there.
Mm-hmm.
It is, that being said, some people have suggested that viruses can effectively, not in the absolute strict sense, they can effectively kind of go extinct. And I told him, I said, extinct is a terrible choice of words.
Yeah.
Don't use that because the other side will not cut you any slack when you use word choices that they can exploit. So, and Dr. Dan's a professional virologist, and I'm just like, okay, Dr. Sanford's a plant geneticist, and also he studied human evolution. And so the whole thing about the viral thing, it's like, okay, you can suggest it, but the other thing is in his book, the 2008 version, he said maybe viruses. He didn't even propose it in the original book, and I said just leave it that way, because his book really was about human evolution, not viral evolution. So what ended up happening is an obscure part of his original book, and then plus the paper, it gets all the focus. And they said, well, if we can refute this part, we can refute genetic entropy's application to human evolution. And I said, no, that's not correct. So you can't. I'm even going to grant, hypothetically, that he may have been wrong about H1N1. I'm not saying one way or the other. You can't use that to refute the problem of genetic entropy at the human level, because there's no major geneticist I know that thinks our genome is improving. So if you want to argue that Dr. Carter and Dr. Sanford were wrong, I'm not going to defend one way or another, because it can't be generalized to the problem of humans.
Yeah. Is it specific to only humans or eukaryotes, or where does it, mammals?
It is. I'll show you what it is specific to. there is this Poisson distribution here. It's dependent on reproduction rates. The number of mutations, let's see, I did a, okay. This is, to answer your question, we have to go through this, and I can walk you through some of this. But basically, the larger your genome, the more damaging mutations, the more mutations per individual per generation. So if you have a small genome, you don't have as many mutations per individual. If you have a large one, you have more. And if the human genome is mostly even reasonably functional, even 10%, that's still 10 mutations per generation per individual in something functional. So this is from peer-reviewed papers here. Let me see if yeah, there's the reference. Let me, this is from Kimura's paper here. So there, I gave a reference here. And so it talks about, and so I kind of did the derivations here to show how we can get that. So this thing called U is the number of mutations per individual per generation. So you're asking, does it apply only to mammals or whatever?
Mm-hmm.
It's more subtle than that. It really, the question is how many mutations per individual per generation. So a small viral genome will not have as many mutations per individual per generation. And so what happens is if U gets very, very large, the question comes, how many, and this is where it gets a little bit hairy, but I'm just going to give you the bottom line. You can go through this. There's the derivation here. I'm just blasting through it just so you know that I walked through it. So
Yeah.
what ends up happening is if the number of mutations per individual per generation, if that U gets large, this tells you how many children each female needs to make so that you can have at least one. You have a probability that at least one child is free of mutations. So with one mutation per individual per generation, each female has to make 5.4 kids just to have one kid that's mutation free. Once you get up to 3, she needs to have 40 kids. And then I specifically cited, let me see if I have it here. There are papers that actually will confirm this, and I have references. But once you get up to like 10, you need 44,000. When you have 82 per individual per generation, the number of kids the female has to have is 10 to the 35th power. And that was also supported by Dan Grauer himself, who hates the ENCODE project. So when you said this is applied only to mammals or whatever, it's not asking the right question. The question is, what situations does it apply where the mutational load problem applies? It applies to systems. So to answer your question, it applies to systems where the number of mutations per individual per generation is too high for the reproduction rate.
Okay.
So if you have plants that can make lots of different kinds of seeds, and I'm not a plant geneticist, then maybe hypothetically you could have lots of offspring per plant. So this is a well-known problem among the elites in population genetics, but your typical evolutionary biologist doesn't understand this. They don't care. And that's where I get a little bit angry is they have one set of researchers claiming one thing and the other set of researchers showing that it's incompatible, and then they still keep saying evolutionary theory is proven. I'm just like, you have all these internal problems you're not dealing with. Dustin, are you here?
I'm here, but I don't know if you can hear me.
I can hear you finally. What'd you do, brother?
You can hear me?
Yeah,
I brute
Hallelujah.
force just repeat, and it finally let me through.
Okay, I'm sorry about the restream problem, and we got StreamYard going. So
Not your fault whatsoever, man. I appreciate you even doing this.
so thank you so much for your
thing.
perseverance. So Jackson, I hope this kind of gives you a little bit more insight to the subtleties of this.
Yeah.
And I
read
don't mess with the viruses. I'm sorry.
Yeah.
That's a Dr. Dan thing, and I'll just assume for the sake of argument that he's right. I'm not conceding that he's right. I'm just saying whatever he has to say about viruses won't apply to humans. This is what applies to humans. And the reason I can say it does is one of the people, one of his heroes here, Dan Grauer, he has essentially the same figures that I come up with my application of the Poisson distribution. Now he's off by a factor of less than an order of magnitude. We're close. He uses 5 times 10 to the 35th power. My number is just 10 to the 35th power. So we're kind of, when you're talking about that many babies per female, it's like, okay, what's another multiple of 5? I don't care.
Yeah. Yeah. I did read something kind of interesting related to genetic entropy, and I came across it by accident. But I was reading that both chimpanzees and humans are acquiring more deleterious mutations than other mammals.
I've got a question.
Sure. So since you fought so hard to get here, is it okay if I take Dustin's question here now?
Yeah.
Okay, so bananas.
I'm adjusting your mic volume here. Okay, go ahead, Dustin.
So bananas have 44% of the human genome. Why don't we see degradation of any kind, such as you see with inbreeding, when you have extreme clones of the same plant over and over again? This presents a huge problem for genetic entropy as well as evolution theory, because you would think with a genetic clone, an exact copy, you would run out of material that would be considered genetic material, right? Or you would run into the same copy and over again until it produces its own specific kind of mutation. We don't see that, or at least I haven't seen any major difference in any kind of bananas.
So I just did a quick Google here, and you guys can see it. I said, what's the size of the banana genome? 523 megabases. The human genome is like 3.3 gigabases. So the human genome is at least 6 times bigger than the banana genome. How can it be 44% similar as a matter of principle?
Yeah,
it can't
that was what.
Not right.
It can't even be 15% because the human genome is bigger. So it depends on how the comparison is done. If we do, and I've often said this, how these comparisons are done, it's like, okay, you take any 2 random novels and you take all the words, you make a dictionary of all the words in one novel and a dictionary of all the words in another, and then you compare the spelling between the 2. You're going to say these novels are like 100% identical or 99% because of the way you're snipping out little pieces and comparing them. And that's why it's an illegitimate comparison. That's how you can get these ridiculous numbers that it's 44% similar. And I'm just pointing that out to the viewers. It depends on how you do the accounting. And I'm just saying that it's illegitimate. Does that add a data point for you, Dustin?
Well, what I'm asking is, where's the genetic mutation rate at for the banana? We don't see the same genetic rate in other life forms, right?
I don't know what the genetic, I don't know if anyone's done an estimate of the rate of mutations in bananas.
Bananas? But,
Yeah.
well, we have
But-
estimated rates in humans, though.
Well, how many per individual? And that's the other thing is how do they actually count individuals in bananas? I'm not a plant person. I'm really, that's another way of saying I can't answer your question on that. I can say that the banana is not immediately relevant to the question of whether humans are deteriorating. That's all I could say. I don't know if
In the
I'm sorry
terms
if I can't be helpful.
in terms of evolution, right? So what I'm getting at is bananas, as far as their reproduction, probably thousands of generations beyond humans, because we reproduce this food to eat, correct? Are you following me?
Yes.
Why aren't we seeing any kind of a genetic mutation in the banana, which is straight cloned, like you would when you see populations of isolated humans being interbred? You see genetic abnormalities, but the banana has no one to mate with. It is a straight clone right off the top of the plant. We
Well,
don't see any genetic.
You won't
I'm
have,
listening.
I don't think that there is much genetic degradation when you have clonal reproduction.
But don't bananas have flowers and seeds and all that?
Yeah, so the plants are special because they can be clonally reproduced or through the cycle of sexual reproduction. And
It's called grafting.
So this is an Emery Moyna question, and I'm so are you trying to say this is a problem for evolutionary theory or genetic
I'm
entropy
saying
theory?
it's a problem in either direction.
Yeah, and all I could say is I can't contribute in anything meaningful way to the question of genetic deterioration in plants. However, John Sanford was a plant geneticist. He said he was in the sexual reproduction cycle. He was seeing a lot of problems with genetic deterioration. That's why clonal
with reproduction, but not with cloning.
Not with cloning. No, that's why you want to clone. That's why you want to clone.
No, I'm right there with you.
I will tell you this. I do know about the grape cycle more. Domesticated grapes have lost 15% of their genome on one of the chromosomes. 15% is a large number. I mean, just think about applying that to humans. That would be devastating. So let's, we have these, yeah. If you did that to humans, it'd be devastating. So I learned that from the dean of my school, and she just threw this out in a cell biology thing, and I passed that data on to John and to Dr. Sanford. He said, "Wow, I didn't know it was that bad." So, it really is bad for one species of plants that I'm aware of. I can dump the data here in the side chat if you're at all curious. There was another question here. Was that you, Jackson?
Yeah. Is there really ever going to come a point where the whole population goes extinct and the genome just can't take it anymore?
They call that mutational meltdown, but there are so many examples where the creature is just like so blown away that extinction isn't really the question. Some people have framed genetic entropy in terms of purely extinction, but the real issue of genetic entropy is if you're having such a net reduction of functionality, how can you, if that seems to be the normal mode of evolution, how can it build complexity? And that's an outstanding problem because all of our observed and experimental evidence is that there's genomic decay. even if there's reproductive fitness increases. And that was the title that I saw of a paper by Lenski himself. And I said that one that shows that the notion of evolutionary fitness is useless because it's not a metric of functional decay. So to your question, do some creatures actually go extinct? I've seen experiments where we've had cell lines go extinct in yeast, and some of the bacterial cell lines I think may have even died in Lenski's experiment. It just kept mutating, and it ended up being a dead end. It just self-destructed. And so everyone's saying, let's keep it going. We might see it just totally self-destruct, the whole lineage. And that would be pretty devastating for evolutionary theory, where fitness gains in the immediate short term lead to destruction of the entire population. And so everything that he's been advertising and hyping up, if they continue the experiment and we continue to see these self-extinctions, it would be actually a pretty devastating result. And so the ID proponents are actually hoping he'll keep these lines going, where he's subjected to such extreme selective pressure that now the genome is fragile. So in the example of human evolution, we're seeing natural selection is actually causing a degradation of human intelligence. Natural selection is actually destroying our genome. It's not helping it. And Dr. Dan was not too happy I was citing all these papers he was contesting. I was like, Dr. Dan, I'm afraid you're in the minority here. People that actually study this are very concerned. And I'm hearing Dapper Dino, and he's my friend. I'm just like, Dapper, you can't. You keep saying you think the humans are increasing intelligence. I said, we have objective evidence. One measure of intelligence that is objective rather than IQ test is the speed of nerve conduction.
There's 7 levels of there's 7 different levels of intelligence, right?
Right, but I mean, rather than tests, we can actually, like written tests or whatever, we can actually measure the performance of the actual nerves. And so very intelligent people have, their nervous system is, the reflexes are very fast, and the way that the nerve signal conducts is fast. It's measurably faster than people that have lower IQs. So I'm trying to go to an objective test. We can, the IQ test can be debated, but you can't just wave away or argue away something as objective as the speed of an electrical signal. That is a measurable change, and it is disconcerting that it keeps going down. It also
isn't that governed by the sacs that are inside of the nerve cell itself that releases the fluid for the transfer of the power, the electricity?
I think
that's basically.
The actual mechanical reasons may not be known because the nervous system is very, very complex. And this is another sad thing to say. It also will increase the incidence of mental illnesses and autism and who knows what else.
Alzheimer's.
Yeah, probably even Alzheimer's. And I shouldn't laugh, but okay, natural selection. Women who are very intelligent don't have as many kids as the less intelligent. That's a known fact. And that will lead to that's an example of natural selection degrading our genome right there.
Do you suspect that an intelligent parent would produce a more intelligent child because of their genes or because of their environment?
It is the genes. And the way I could say that is that's why the nerve conduction speed studies are very important. That's something that's independent of the environment that can be, I mean, I'm sure if you're taking drugs or whatever, you could affect your nervous system, but I'm just saying that's something that you could measure genetically. Like say at a young age, you could take their nervous system cells, or you could do nerve conduction studies. So, a person will have, I mean, he might be able to improve his reflexes up to a limit. but you sort of are, it's just like physical speed for athletes. They can develop it, but that
Reflexivity.
there's some people that have more natural talent. And I know it's not been politically correct to talk about genetics affecting intelligence, but I'm trying to talk about objective facts we have to deal with, because if loss of intelligence is heritable and then also mental illness, that's a fact we're going to have to deal with as a society. If we're starting to slowly lose our minds, it's sad. And that's what I've been trying to, dumber equals more evolved. But that's why I mean, just on a philosophical and religious, if the human race is dying, that should be very sobering how we live our lives. That's it's certainly the Christian message that the world is broken and we're in need of a savior, and that kind of brings it home more. And if that's not enough, we expect the solar system to die at some point. So, what do we actually live for at this point if humanity and civilization will end? So that's me waxing philosophical.
Philosophical, I agree.
Sorry, go ahead.
No, you go ahead. You're fine.
Okay. I was reading something about in the past evidence of whole genome duplications in certain animal species or plant species. Have you heard anything about that?
Yes, there's definitely whole genome duplication in plants, and that's why I wish Emery were here. He's our plant guy. He studies what they call polyploidy. So that's where you do have whole genome duplications and polyploids. And it's a little funny in humans. In the germline, there's not whole genome duplications, but you do have it in somatic cells, like the myocytes, the cardiac myocytes. You have sometimes aploidy. There are 8 copies of the genome in some cells in your heart when you're developing from like in childhood, which is kind of interesting. We don't know why it does that. It seems to be a valuable thing, but then why do the other cells in your, only have 1 copy of the genome, but your cardiac myocytes have 8 copies, 12 copies, huge numbers of copies.
Isn't that in the cell division in the cleavage flow?
I don't know the mechanics of it. I don't know the mechanics of why it does that.
Well, that's how they theorized that autism was started. Was that the genes were unevenly split in the cells?
I never heard that. So I'm sorry. If you all want to talk a little bit, I'm looking up that reference in the grapes. Since you talked about bananas, I did want to put one on the grapes because I'm familiar with that and the 15% loss.
Yeah, I would like to see that, because as far as I understand with bananas, you don't see that. And I'm no more than a novice at any of this, but I can pretty much follow along.
Yeah, I was reading that primates, especially chimps and humans, are acquiring more deleterious mutations than other mammals for some kind of short-term gain or something. I don't remember the details.
Well, and the thing is that if you're seeing a result like deleterious mutations, why don't we see the same result in animals and plants? The genomes are the same. The argument from evolution is life all comes from a similar source. Why doesn't why are certain genetic traits inherent in humans if the four base pair are the same among all plants? Now, that's not to say that there isn't disease in plants, but it's more prevalent in humans. So there's some kind of a disconnect there, some kind of I'm having a hard time understanding that.
Well, everything has a different genome and mutation rate, genome size. Like
But they all came from the same time,
same organism originally, yeah.
right?
Okay, so I'm going to
So
try to copy. And I can't. Let me try to grab it from another spot, and then I'll read from it. Let me see if I can find the title of this. A toast to the genetic diversity of grapes. Okay. I'm gonna. I'll just read a little bit about it because I don't know that if we had this happen to humans, I don't know that we could. I don't think we could tolerate that amount of change. At the very least, this would be kind of scary if it happened to a human. So
one question, if before you start reading it, you can keep an eye on this. Is there any difference in flavor in these mutated grapes? Is there any difference in size, shape?
Absolutely, absolutely.
Okay.
I mean that's a key feature of how we have different varieties. So maybe this will answer your question. And greetings, Danish debater. I had two guests here on another channel that just wanted to ask me questions, so that's why it's random. We're talking about specifically genetic entropy, and that's where we are. Honestly, I am worried about the world situation. I've been praying a lot, and I'm just trying to get my mind off things because all that's happening in Ukraine, it could escalate. It could escalate very quickly. And anyway, we're talking also about solar system evolution. But so let me talk about the grapes. Here is a discovery well worth toasting. A research team led by Professor Brandon Gaut with the University of California, Irvine, and Professor Dario Cantu with the University of California. Davis has deciphered the genome of Chardonnay grape. By doing so, they uncovered something fascinating. Grapes inherit different numbers of genes from their mothers and fathers. This paper has been published in Nature Plants. So this was referred to me by the dean of my school, and she was my professor of cell biology. And so this was worth almost taking the class for, just that she gave me this popular version of interest to genetic entropy. The team devoted 3 years of study to what are known as structural variants, or chromosome changes, in the genomes of Chardonnay and Cabernet Sauvignon grapes to determine their genetic similarity. Each of the fruits has about 37,000 genes, which is amazing because humans have about 20,000, so the plant may actually have more. Each of us inherits one copy of their gene from their mother and one from their father, says Professor Gaut. One would assume that the grapes inherit 2 copies of every gene, 2, with one coming from each of the 2 parents. However, we found there was just 1 copy, not 2, for 15% of the genes in Chardonnay. And it was also true of Cabernet Sauvignon grapes. Together, that means that grape varieties differ in the presence or absence of thousands of genes. These genetic differences probably contribute to many of the difference in taste between wines from different grape varieties, said Professor Cantu, and they definitely contribute to one important feature of grapes, their color. So I hope that answers some questions there about the loss of genes contributing to the variety. 15% is a large number. That's just tremendous. I can't imagine humans being able to tolerate that much loss of genes.
Humans are more complex as far as needing oxygen and such. So
Yeah, we're not very tolerant. Well, not that we would ever want to experiment on humans, because that would be there'd be ethical problems to be knocking out genes, and uh, y- you'll induce birth defects in a fellow human. That's just unthinkable.
well,
But
but then here's the question: at what point would you be able to call that devolution?
yeah, yeah. So I would call it's starting to come into they don't like to use the word devolution. They use the word reductive evolution. Reductive, because you can when you have a gene loss, that is a loss of a functional unit. Um, there's some other aspects of what you call devolution that may not be a total gene loss where the gene is compromised, and that gets into some subtle difficulties. But biologists generally will say when you lose a gene, you've reduced the genome, and we call that reductive evolution. So that is an official term that is used. It's usually used for microbes, but there's no reason we can't expand it to things, eukaryotic organisms, like plants and animals.
What about like de novo gene birth, new genes arising?
Um, there are new. It depends on what you mean by a new gene. So
Is it a sequence like the 4-base pair that's different? Jackson?
you can use. Okay, let me just say this. At what point is a book a different book if you just change one letter? So how can we objectively measure that we actually have a substantially new gene? And I tried to cover that in one of the shows on major protein families. One way that this can be objectively measured is how new it is. If you have a de novo gene that looks like its parent, it's like, okay, you've not really made a new architecture. And I have pictures that can kind of illustrate this if you give me a moment. Because we don't have de novo of major protein families. And so how do we objectively say this is majorly different? And the objective way is to use these phylogenetic alignment algorithms. And even Dr. Dan was agreeing with me. He said he can't align these. So if you like take that's why we could say we could see a topoisomerase in a human, and we can also see it in a plant. They're similar enough. If I grab this, you'll see it pictorially what I mean by de novo. So I'm going to have to try to answer your question, Jackson, by showing a picture, and it'll make more sense.
Okay.
What I'm talking about. Just give me a moment. So if you guys can talk amongst yourself, then
So
it'll give me a chance. By the way, I'm really glad we're talking here, because I can't get a word in over at that dumpster fire there, and I really lost my temper with Mark. I mean, I was livid. He said no biologist said that. I kept repeating. I said he was just dismissing me. I lost it with him because I had paper. I've researched this, and he's just going to dismiss it. And then he said, "Oh, I can't read it on my phone." It's like, well, that's not an excuse for that. You have no justification for saying no biologist has said this just because you can't read it on your phone. All that means is you haven't seen it. It doesn't mean that it doesn't exist.
You're physics, right? You're a physics professor, or your specialty's in physics?
Biophysics. So biophysics is biology and physics. But I'm not a professor. I am a researcher. I work for a geneticist. So biophysics are a special animal. They combine physics and biology. Because the reason for that is there's a lot of, to be able to understand biology, there's a lot of physics now that's needed to understand it. A real good example, a basic example, is geometric optics. So the way that the eye works, you actually need physics to understand, like all the way light is able to get processed, so that
the spectrum, the band, and the way that absolutely.
it is not trivial. And there are other areas of biophysics, like in protein folding and how proteins work. And that's why I got livid at Mark. I'm just like, dude, you freaking don't know what you're talking about, and you won't even let me get a sentence in. And you say, explain to me in one sentence. I'm just like, I just like, I don't want to deal with stupidity of that level. I just lose it.
I mean, I'm not one to bash a man when he's not here to defend himself, but I understand what you're saying, and I agree with you.
And I couldn't get a word in. I'm just like, I give up. And so someone advocated, I don't know, Cheryl, God bless you. Cheryl was saying, LPP, can you let Sal finish his point? And I wasn't going to talk to other people.
I saw that. I was like, so Jackson.
Yeah.
Can you explain to me the concept of vestigial organisms?
Vestigial organs?
Organs, whatever.
Yeah. Just an organ that doesn't do what its original purpose was, really. Like the like Basilosaurus hind legs.
So at what point does the predecessor to the whale, I can't pronounce the name, Optiosaurus or something, Optiosaurus?
I don't know.
Hold on, let me look it up real quick. I don't know. Anyway, at what point does the lower legs of the predecessor to the whale become vestigial?
It probably became what you would call vestigial at the point of probably Georgicetus, when the pelvis disconnected from the spinal column.
So
in a
then
sense,
what the issue that I'm having with this is, at some point the vestigial organism or the organ, whatever, has to serve one function and transition to another, or it's completely useless and then it's discarded.
Yeah, and that's nearly what happened with the whale legs. They're still used for reproduction, though.
So, in equivalence, right? Can we say, for example, I don't even know how I would equate. See, I'm having a hard time understanding how in evolution a whale goes from land to water, stays mammal, but we don't see. I don't know. I'm having a hard time wording this.
Don't see what.
I'm having. I can see what I want to say in my head, but I'm not. The words aren't there. Okay. So okay, I know how I can do this. Okay, so at about what time in the human evolution did the whale evolve from its predecessor? Can you?
Well, that was about.
Can you put it?
What time in human evolution?
Yes, that the whale evolved from its predecessor.
They were just pan primates at that point, 40 million years, 50 million years ago.
Can you say that the rate of change between the whale and the monkeys, for lack of a better word, would be at the same rate?
The rate of change?
Yes.
Not necessarily. It varies. Seems to vary.
And what are the selective pressures that makes the difference?
Could be anything. Could be just usually it's a new environment or something like that.
So, in other words, a whale can go from land to sea without any directional pressure, for example, because evolution doesn't know what the end goal is. And the rates are vastly different.
Well, in the case of the whale going from land to water, there would be a bigger selection pressure to adapt to that environment. So it's not just one generation producing something that's aquatic. It's a slow process, but it would be faster than, say, a primate going to another land, basically land living primate, if that makes any sense.
So in other words, as far as evolution is concerned, whales are thousands of years ahead of us in evolution.
Well, if you want to put it that way, they're ahead of us, yeah. In evolution, I guess you could say that.
But then you turn around and you look at the other end of the equation. If you were like an accountant and you're saying that our mental capabilities would change that rate of evolution, correct? Because back then we weren't driving cars.
No, we weren't.
So where's the equivalent? That's the one thing I've never really understood, right? I mean, if I'm to understand that all life evolved, right? Why do certain organisms get these huge jumps in evolution and leave the other life forms all behind?
Again, it depends on the selection pressure. They vary. Like I guess you want to say severity of the pressure. That's the best way I understand it.
So the severity of the pressure can take 1,000 years of evolution and cram it into 100.
It wouldn't be that fast.
Well, then why then I'm having a hard time understanding how the whale jumps thousands of years of evolution from land to water, and monkeys haven't achieved flight aside from mechanical means. You know, there's no real actual evolution as far as humans are concerned.
Well, there is some evolution going on from the pan primate to the human over the course of
Hey guys.
years. Yeah.
Sorry to interrupt. We have a question from Danish Debater. Can you state your position briefly, like evolutionist, creationist?
Dustin, you can go first if you want.
I guess the best way I can put it, I have my own personal beliefs, but my position as it stands for officially is agnostic.
Yeah, I would say I'm pretty much an atheist, evolutionist, but I'm open to the possibility of a God. So really, agnostic, atheist, kind of interchangeable, I guess.
Oh, thank you. I tried to adjust the mic volumes a little bit. You can carry on. We have a couple outstanding questions, one from Jackson about de novo genes, and then one from Crimson Air. So whenever you guys want me to get around to answering those questions, just let me know. Otherwise, you all can keep chatting.
Do you want to set this up right now as a debate? You want to go for structure or form?
What?
No, I'm here to serve you guys.
Yeah, I'm talking to Jackson.
Oh, I'm sorry.
I'm asking him if he wants to go for structure or form.
What do you mean structure or form?
Well, we can structure our debate around rules, or we can just wing it on form, like good form, standing principles between two gentlemen in a discussion.
I don't know. We can just talk about stuff, but I'm kind of tired for a debate.
Copy that.
I just had a debate, you know.
Yep.
Yeah.
Okay. So I'm trying to understand your position. I really do. I want to get into the way you think. So the thing that I'm having a problem with is that we see thousands of years of evolution in a whale. Maybe millions of years have gone by without any real evolutionary advantage to mankind. And bananas with 40% of the genome, and there's no mutations.
Well, humans have existed for about 300,000 years.
But we were something before we were that, though.
Right.
So then you can't say that we've only existed that long.
Well, no. By that metric, we've existed for 3-point-something billion years.
So then why do we see huge leaps of evolution for the whale and billions of years and no changes in the bananas?
Well, bananas haven't existed forever either, but really, I guess I don't have a great answer for you.
Right. I mean, the rates of evolution between this genetic, whatever you want to call it, I mean, eukaryote, I guess. But the sad thing is, if they're all eukaryotes, then there's no difference between any of the organisms at all.
Hey, guys.
Yeah.
Yes, sir.
Could I weigh in a little bit, because I'd
Absolutely.
like to answer Jackson's question. Would that be okay with you, Dustin? And I also try to answer Crimson
No problem.
Air's question. So Crimson Air was asking about the genetic code, and there's let me just try to explain the genetic code. So we have DNA, the letters A, C, you can see it here, A, T, C, A, G, or A, whatever order. And
I'm not getting any of that.
Okay, this is going to be, this is just, huh?
I can't see any of it, but it's okay. I'm listening to you.
Can you see it, Jackson?
I can see it, yeah.
Okay. So, and let me make sure that, guys in the chat, if you can't see my screen, this is the table of the genetic code here. And so Crimson Air is asking about the codons. So we have 4 letters of DNA, and a codon is constructed of 3 DNA letters. And we only have 20 amino acids. So for example, the amino acid valine here, there is 4 ways to spell valine, as GTT, GTC, GTA, GTG. And I think there is one, okay, arginine has 6 spellings. There's 6 ways you could spell arginine. And so there's 6 different codons that will make the, translate into arginine. So now going back to some basics here. So let me try to go back to some basics here, because this will now answer Jackson's question, I hope, a little bit about de novo genes. So we have 20 amino acids, but then we have 64 codons. There are various ways. As I said, the arginine has 6 ways, 6 different DNAs that can make the same, that will result in the same amino acid. So I hope that sort of answers your question a little bit. It's not the DNA strand. It's the codon can have up to 6 different ways to spell the same, to translate to the same amino acid. And they call that codon degeneracy. But we have 20 amino acids, canonical. There's some extras like citrulline or something, but this is like the more basic, the simplistic view. And it's close enough for our purposes. And so you have a protein here like collagen. So we have a protein like collagen. You can even buy the collagens in the beauty store. But about 30%, 20 to 30% of our protein weight are collagens. And what you can do is you could take the collagen. There are many collagen types of collagen in us, but this is one form of collagen. And you could spell the amino acids in the collagen like this. This is one way to represent it using English alphabetic letters. Everyone okay with that so far? This is the
Yeah, I
spelling
follow.
of, I'm sorry?
Yeah, I follow.
Okay, so this is going to lead to the de novo question. What do we mean by de novo? So there is a pattern here, and I'll highlight it, that's very distinctive for collagen. There it is. Do you see at some point, every third amino acid is a G, it's a glycine. Very distinctive right there. Very distinctive signature of human collagen, of any collagen. And so we could just roughly say the motif is just G and then 2 other amino acids. Everyone okay with that so far?
Yeah.
Okay. Now here's the human zinc finger protein, and I'll expand it. Do you see a pattern there? This is a very distinctive pattern. In that we don't have, most proteins we can't do this for. I picked these 2 because you could actually visually see the issues. So you could see then, and we can also, if we group it according to this Venn diagram, you can even see even more patterns. So this pattern isn't an accident of our perception. It's a real thing. It's what they would call a violation of the law of large numbers, that you could get these patterns.
Okay.
So you could see the difficulty of evolving. I'm getting a kind of funny sound here. I don't know which one it's coming from. So the difficulty of evolving, let's say, collagen from a zinc finger or vice versa, and then also imagining how difficult it would be to evolve it from a common ancestor. They have radically different structures. Now, that being one way to imagine this, is you have a different architecture for a piano versus a car versus a helicopter. And
There's
I had
a
another one.
KA-52.
I'm sorry?
That's a KA-52 up in the right corner. Russian helicopter.
I didn't know that. Thank you.
Yeah.
Hey, and I was in the military aviation industry, and I didn't know that. I know some of the planes. I specialized in the F-18 and the A-6 Intruder.
Oh, yeah.
Yeah, that was me in another life. So this does pose a little bit of, this is what I mean by de novo genes. So we can say, like, if we have like the zinc finger in a human, we find one in a pig, and they're spelled differently. So the human is only, between humans and pigs, the zinc finger 136 is 50 to 60% similar, depending on what comparison algorithm you use. But you could see that they're similar. But the spelling of the letters, like between them, you could see they're not exactly spelled the same, but they have kind of the same major characteristics. So like, for example, in this row here, the first row, you could see between the 2 H's, between the 2 histidines, you have IIT, whereas in the pig it's ESS. So if you just vary this, is this a de novo gene between the humans and pigs? And I'm like, well, they're spelled differently, but it's still the same basic architecture. So this is where I was getting to, that it becomes subtle. What do you mean by de novo? Because it's very, if you're just going to change one letter, it's like, well, yeah, you could say that's de novo. But what I mean by de novo would be a majorly different architecture, like say, like a collagen. And what we see in major protein families is you don't get a major development. You don't get de novo in terms of a major new architecture unless there's some gigantic accident. Simultaneous point mutations, or what they call translocations. Now we can also do what we did with the collagens. So the collagen between human and zebrafish, you can see that has the same pattern, but they are only 75% similar depending on your scoring method. So you could say, yeah, de novo in the sense that you might be able to take a human and you can mutate a few of them, and you could sort of say it's de novo in the fish or vice versa, but it's not really the same major architectural change. So these little kind of, to use the term very, very, very loosely, these kind of microevolutionary changes are not a good explanation for how you have these major architectures. You can't extrapolate it. So that's what I say when I'm talking to evolutionary biologists. You can't generalize a small change to make a major change. And that's how I kind of describe it, because you'll actually get a very, very different behavior. Whereas we've experimentally shown that, like, for example, when we have this one, let's say you have a collagen in a human and a collagen in a fish. I can't speak for collagens, but I can say for the enzyme that I study, the topoisomerase, you can take a human topoisomerase and insert it into a yeast, and the yeast will live. But you can't take a collagen and then replace the yeast topoisomerases with collagens and expect the yeast to live. It'll be lethal. So that's why I'm just pointing out, Jackson, what one means by de novo. It needs a lot of qualification.
Yeah.
De novo, as far as this, they're not good explanations for de novo as far as like between these major architectures.
Okay.
And that is, I would say that's a deal breaker for naturalistic evolution. You can still assume common ancestry, but the statistical difficulty of creating functional, you could see that there's not going to be, it's difficult to make a functional intermediate between these two. Because I could tell you, if you start to break, like say these individual zinc fingers here, that could be just very damaging to function. And then same here, if you start to mess around with kind of these very specific locations, it could be very, very detrimental to the organism, so that you can't have a gradual change from one to the other without causing a lot of damage. And then, of course, the evolutionists will say, what about gene duplication? I said, you're going to have also problems there. It's a little bit more subtle. But you can't expect to have selection pressure. What ends up happening is natural selection would actually prevent change rather than facilitate it. Because if I start mutating this to try to make a collagen, it'll end up being a bad gene that's just like useless and should be eliminated. So actually natural selection will prevent evolution of new protein forms. Major, I'm not talking about small, but major architectures. So thanks for giving me the floor there to explain my views.
Yeah, it's interesting. I've another kind of observation about genetic entropy. You were talking about we're talking about huge selection pressures on like grapes and the E. coli and the Linsky experiments. And could it be that things under these huge unnatural selection pressures exerted by man are somehow going through reductive evolution, as you say, faster than the background, and it's causing these strange effects?
Anytime there's strong selection pressure, let me try to get your question right. Are you saying whenever there's strong selection pressures, does it cause reductive evolution?
like abnormally strong, like with
Yes. Short
selection,
answer, yes. Short
Yeah.
answer, yes. It tends to cause reductive evolution. And it's also, there's a phenomenon called interference selection. So let's say you have an individual that's fast and an individual that's smart. And the strong selection pressure is a lion. So intelligence would be considered a nice trait, and so would speed. So let's say you have the fast, dumb individual and then the smart, slow individual. The lion comes along and devours the smart, slow individual. And so there you have would be a very good trait to have, which is intelligence, and he gets wiped out by the strong selection pressure that the lion. So that's an example of why it induces reductive evolution, is because you have an otherwise good trait being just kind of squashed.
kind of
Because
like with like what I was talking with Dustin, the selection pressure of the whales to lose their hind limbs in a similar way, in a not exactly the exact same way, but
yeah, I mean, let's just, for the sake of argument, let's say that there's strong selection pressure to lose. So you can use selection to reduce, but it's very hard to construct because you need the right mutations. You can't select for something that doesn't exist. So like, it becomes very apparent, it's hard. It's easy to kind of maybe guess and imagine that it could evolve, these new traits when you just look at the
Question.
fossil record. But when you look at the actual molecules and the individual proteins and the cellular changes,
Question.
the required coordination is just brutally evident.
yeah.
Okay, I have a question. Okay, so we've established that there was a strong selection pressure on the mammal that is whale. What is the strong selection pressure that would select only the mammal whale and force it back into the ocean when only, I don't know, depending on what time period we're talking about, millions of years they had just crawled out of the ocean?
Well, between the time where the Alpistus stigalians of the Devonian period, like 380 million years ago, to the whale, which is 50 million years ago, it's quite a gap.
Well, but here's my question. The gap is irrelevant because we had other mammals that thrived on the land.
Right.
So if you're saying that there was a selection pressure for this specific whale, forcing it back into the ocean, why did it only select the whale? Why didn't it select any of the other mammals that were around it? Why just the whale? And it was a severe jump in evolution from land to ocean. I think we can all agree about that. Why was it only the whale?
Why only the whale? Well, could just be one species took a more semi-aquatic lifestyle to get some food resource, something like that.
So for thousands of years, mammals have been crawling their way off of the ocean to get to a better food supply, for only the whales to turn around and decide that this is a bad idea.
I'm not sure what you're asking.
So, okay, mammals originated in the ocean, correct?
Well, not mammals didn't originate in the ocean. The tetrapods did.
The tetrapods that split off and decided that it was better on the land, right? Up until the whale evolved, and then the whale decided, I don't like it here, and I decided to go back to the ocean. There was a strong selective pressure that selected only the whale out of all the other mammals and kicked it right back to the ocean.
Well, Sirenians and pinnipeds, they've all kind of started the same process of going to back to the water, so to speak. And like Sirenians, like Pasosiren, again was a four-legged, basically manatee, and also about 50 million years ago. So it evolved relatively rapidly to a new environment.
So then, what selective pressures selected those mammals but left humans alone?
The radically
We're all mammals,
right, but it would be a radically different environment.
but we were all alive at the same time. It's not a different environment if we're all if the eukaryotes that we evolved from were alive at that time. Why didn't those eukaryotes get forced back into the ocean with the other strong selective pressures?
Well, there isn't just one ecosystem on Earth or one environment. Oh, he's gone. Sal, can you hear me?
I can hear you guys. I wasn't paying attention to the what was going on. I was typing in the chat to Crimson Air.
Oh, okay.
We lost
Yeah,
Dustin.
lost
Oh well. Huh?
Yeah, we lost him.
Yeah. So I don't. Yeah, technical problems happen. So Crimson Air is asking. He's asking me some tough questions here, and I tried my best to answer them. And sometimes a question is, it's like I'm trying to guess what he's trying to say, because there's some questions that are a little bit like I either never thought of it that way, or whether it's relevant to the creation evolution controversy.
Yeah.
Or whether it's a question I can make sense of, but I'm trying my best. So I hope I answered that. So Dustin, you're back? Oh, can't hear you, Dustin, if you're trying to talk.
Sal, I have another observation. Can you hear me?
Yes.
Okay. So this fast evolution seems to be the problem when it comes to the genome. I think if what we're talking about is correct. So the deep timeline kind of makes sense in that regard.
So you're talking about rapid evolution?
No, I'm talking
sorry.
about very slow evolution.
Various what?
Very slow evolution.
I've never heard of that. Various slow?
Very slow.
Oh, very slow evolution.
Yeah.
Okay.
Like with the example of the whale, that happened very quickly, and there was apparently some kind of reductive nature going on. But I don't know. What I'm trying to say. I kind of lost my train of thought. But
oh, you can get okay. The fast evolution you can get is definitely reductive, because it's very easy. It's easy, as even Michael Lynch, an evolutionary biologist, said, it's easier to break something than to make something.
Yeah, that's what I'm saying. Once you start going faster, you get more of the reductive evolution than if you go.
Oh, absolutely.
Yeah,
absolutely. It's easier to lose than to make. It's easier to break than to make. You kind of see that, like, sorry to talk about war, but yeah, it takes a lot longer to build a building than to blow it up.
Yeah.
And same could be said about genomes too.
So what I'm telling you is, I think deep time is the savior to your genetic entropy problem.
I'm sorry?
I think deep time is a savior to the genetic entropy problem.
Deep time is a savior for genetic entropy.
Mm-hmm.
Is that what you said? No, genetic entropy, the way to frame it is loss of function. That's the more correct formulation, rather than extinction. I've tried to advocate for that, and with the author would be John Sanford. And I don't know that I've totally persuaded him that's the better formulation, but it's the more defensible one.
Yeah.
If we refer to genetic entropy as loss of function, and loss of function, at least, again, function is kind of a metaphysical term. What I mean by that is we can objectively measure like things like mass, weight, energy, temperature. Function is kind of a little hard to define, but there's kind of a universally agreed viewpoint that if you lose a gene, you've lost a function. You've lost something that you could use at some point. So reductive evolution actually is a form of genetic entropy. It supports the overall picture that biology naturally loses functionality. It doesn't construct it. If that's the net direction of evolution that we observe both experimentally and observationally, then how can we justify that evolutionary theory is correct without invoking miracles along the way? Or
what I'm saying is, let me try to explain. In the vast amount of time, most organisms don't seem to change that much. It's only when they're subjected to a new environment or some other major pressure, they go through a change over, say, a relatively brief period, like the whale me and Dustin were talking about.
Yeah.
And,
so the question is, if you need constructive change, whether selection can affect it. And just for me, I don't believe it's going to happen.
Yeah.
And that's why I don't believe in evolution.
Yeah.
Evolution of complexity, for one. So I can grant hypothetically, just for the sake of argument, common ancestry. But then even granting that, it seems you would need miracles to transform a land creature into a whale. But I don't go there because it's easier to argue at the molecular level where it's so clear. See pictures like this. Pictures like this make the problem really brutally apparent. You know, I can say that. How can you have selection pressure to convert one from the other, or what did the common ancestor look like, so that selection could build these two from a common ancestor? And even as a matter of principle, that I don't see how that could be sensible.
Yeah.
This is why I focus on the molecular side, because then we don't get into such speculative arguments. You can actually see it visually. But it's an easier way to state the problem.
Yeah.
That's why you don't hear me talk a lot about the fossil record. Because we're dealing with bones and shapes, whereas these are discrete. These things you can are more subject to computation about the difficulty of making one from the other or from a common ancestor.
Okay.
So what I mean by common ancestor, so like let's say we had this one here. You have the fish version of collagen and the human. You can kind of construct a hypothetical common ancestor of what it might look like for that particular collagen. And you could say, well, it probably had a lot of the same glycines here and a few of the other parts. And then, so the ancestor looked something a little bit like the human and something a little bit like the fish. And that's believable for that protein. But where you get into problems is what's the ancestor of the collagen and the zinc finger? It's practically impossible to characterize the common ancestor there.
Yeah.
Can I take a question from Crimson Air? He's probably been, while we're trying to get Dustin back here.
Yeah.
If I understand correctly, all codons and proteins are useful in the same way that the letters of the alphabet are useful, but the higher function depends on their arrangement. Not quite. all codons can make either an amino acid or a stop codon. Talking about utility and usefulness, I don't want to get into that realm until we talk about specifics. I'd say most proteins are useful, but some are life critical, and that's the important thing. So I sympathize with you asking these questions, but I'm trying to frame that. I'm trying to put on the table the questions that are that evolutionary biologists need to deal with. And so I'd say all codons have a use. We could tell that the ribosome is built to process all those codons. When I showed the genetic table here, when I showed this this table of the genetic code, so they have a use. And I'd say most proteins are useful, some are life critical. So for the codon is like letters in the alphabet because it does translate to a specific letter of the amino acid. And those letters I highlighted here earlier. So the alphabet, the codons will result in, let me see if I have you can even see the correspondence with the English alphabet here. So the codons do end up translating, the ones that aren't stop codons, translating into a, they start translating into letters that you can correspond to some English alphabetic letters. But the proteins I wouldn't say are alphabets. Individual proteins, or the more formal term would be polypeptide, those are more like entire words or functioning paragraphs. The analogy sort of breaks down there whether a protein's a sentence, a word, or a paragraph. I'm not going to go there. So I hope that's helpful. Dustin, are you back? Dustin? Oh my, he's having some connection issues. And we lost him again.
Yeah.
Poor guy.
Yeah,
if there's some other questions I haven't addressed in the chat, just retype them. I can't. I'm not multitasking well, and we'll have to shut down soon because it's getting late. So,
yeah.
And I don't have unlimited hours like LPP on StreamYard. I do have it on Restream. So Jackson, if you had some questions, and then Dustin, if you have some, then we'll have to wrap it up.
No, I just think that was an interesting conversation. I'm glad you had me on.
Oh, thank you. So Danish debater, could you use the Dustin downtime to reply to the half-life queue? I didn't see, what's the half-life queue? Someone probably put it earlier and I've not seen it. Okay, so there's we're after 2 times half-line. There should be nothing left. The Yek has claimed as much. I've never heard that. Okay, if you have okay, so I'll just go to something I'm aware of. Carbon-14. If you have, say, 20 half-life cycles, so the half-life of carbon-14 is 5,730 years. 20 half-life cycles, and let me just put that through my calculator here, is 114,600 years. After about 20 half-life cycles, we'd have a very hard time getting detectable amounts. That's about all I could say. I wouldn't say 2 half-life cycles for carbon-14, more like 20. Now, if you speed things up, if you double the speed, I don't know that you would. I don't know that that works either to eliminate any trace of carbon-14. So I've never heard a young Earth creationist talk about that. It was a reply to Rebecca's video about her video. Yeah, I think that's. I watched that video, and I both the original and Rebecca's response, and I would be very confused about hearing it framed in that way. So thank you for the question. I'm really glad you all showed up. Dustin, is your audio working? Oh man.
Guess not.
I guess the solution is to go back to LPPs and see if you can connect better there, but then you'll have to be in a shouting match to try to get airtime. So
I bet LPP's gone to bed by now.
Huh?
He's probably gone to bed by now. Probably can't get in.
Yeah. Oh, SFB mod. I banned him from the channel because I'd like, I want to be on friendly terms with him, but he went after me once, and then some of the stuff he says, that just doesn't make sense. I don't think when a question is so convoluted, I don't answer it. So what's an example of a question that's so convoluted? If I said in Euclidean geometry that there exists square circles, and then you try to start asking the properties of square circles, that's like a question that's so badly formed it's not even worth trying to answer. It's too convoluted. So yeah, I know him, and I've had to ban him from the channel because he misrepresented me in a really bad way, and I'm like, I just don't need to deal with that. Some of his comments in the side chat were just so confusing, and it's more like drivel, and I'm sorry to say that, SFB mod. So I'm glad I could be of service to talk about my viewpoints and then also talk about basic science. And I'm really sorry I have to close this down, but it's really late. And people know I have sleep issues, and I just end up being a zombie during the regular day because I don't know what it is about my circadian rhythms that I'm just awake at the most inconvenient times, because I really did want to be asleep right now, but you can see I'm wide awake.
Yeah.
So Jackson, it's really great finally having you on the channel, and we talked about it. And I hope I've been helpful, at least enlightening you on what my viewpoints are.
Yeah. We will have to talk about the fossil record someday, though.
Yeah. Yeah. And then, I'll be having streams on solar system evolution.
Yeah.
And also atheists who became theists. And Dustin, man, I'm really sorry that we're having connection problems. So thank you for joining us, Dustin. And I'm praying the world situation will not get out of hand. I'm not trying to take, I'm very concerned, even before nuclear war, that a cyber attack could be really bad for our financial situation.
The Ukrainian...
I don't think people realize how just if anyone remembers the meltdown of 2008, it won't. It's like cutting a nerve when you cut the transatlantic cable. If you can't do banking, stuff can go really bad very fast. And
that's probably the angle they'll take instead of a nuclear war.
I'm sorry?
It's probably the angle they'll go for instead of nuclear war.
Yeah, it could be almost as devastating, at least in the short term. Before the electronics and all, we weren't so dependent on technology, we wouldn't even worry about it. It'd be like a telegraph line being cut. people are still able to live. But this cutting the transatlantic cable, because we're so dependent on being able to do international banking, what it could do to the monetary system, we hate to think that there could be no civilization as we know it. And I know that may sound like hype, but I studied the financial markets, and I'm just like, we were lucky to survive 2008 with the derivatives meltdown, but everything can just spiral out of control, and it's scary. So
Yeah.
This is the time to be praying, guys. So Jackson, Dustin, everyone in the side chat, it was great, and it's really hard to say goodbye, but Lord willing, we'll talk again. Take care and have a good evening.